Thank you very much, Seth. I was very excited when you sent me that invitation, and I'm very, very happy to be here. I've known some of you for a few years now. In fact, I was at Winchester at the LIF 11 almost 4 years ago. It was an excellent conference, and so I met Jason and Richard and you and other people, and then we met again. And during ICA, there was in between there was Denmark, LIF 12, and so on. But it's the first time I'm here, so I'm really glad that it finally happened, and especially I was intrigued, as I told you, by this complexity center here, which is relatively new, which is complex systems science, generally speaking, but yet at the same time is also has this, I think, unique focus on what simulation is in the name of it, and focus on computer science and bio-inspired computer science and LIF. I like the fact that the Complex Systems Center has a focus on artificial life and trying to bridge engineering and science of complex systems. And I'm going to try to entertain you with thoughts about these bridges between looking at complex systems, natural complex systems, understanding how they work, made of agents, interacting locally, emergent behavior, and so on. And on the other hand, wondering how we can create our own complex systems and for what purpose, and how we can be inspired by the one we look at and to create new ones, or maybe tamper with the existing ones, as in synthetic biology, which is briefly illustrated here, and create hybrid systems. So this back and forth between observing and understanding on the one hand the science of complex systems and designing, or not too much top-down design, of course, but designing in a certain way, designing the agents of complex systems to create new phenomena that don't exist, to make birds flock differently, to make bacteria cluster differently, do things they don't do usually. And in fact, I like Seth's most recent work about wasp mites, to make wasp or termites do different things, and virtual words, so life as it could be, which is the definition of life. So without further ado, I'm going to dive into this, and the title will become a little clearer as I go. I'm going to spend some time on some general introduction and concepts, and then I will show you, I try to really cut down my introduction to not too many slides, so I will wave my hands a lot, but then I will show you more concrete examples, studies, models I have been doing with my students of the principles I will talk about. So, in fact, let me give you a very brief tour of the plan. Probably I won't have enough time to do everything, but I will first, in fact, start with a model of biology, so biomodeling, a model of embryogenic development, embryogenesis. In that case, the zebrafish by my student Julien Delille, of course, co-supervised by a real biologist, a developmental biologist, Nadine Pairas. He just finished a few weeks ago and defended. Then I will transition to Bioinspired engineering. So this is life, and this is life, all right? And how we have, in this case, not real embryos or creatures, but fictitious ones. This is 3D Evo-Devo, or in fact more Devo than Evo for now, but especially Devo, robotics, creatures that develop and then function in some space. Same inspiration here, except instead of a 3D space, we have a 2D plan, and this is more inspired by plant growth. In that case, maybe networks. The idea is, even if its applications are a bit far away, can we have self-organizing, constructing networks for then purposes of technology building and so on. So the space is different from this one, but the idea is the same. And then, in fact, possibly back to biology, but via a different path, and back to, as I was saying, tampering with biology. Synthetic biology, of course, is the big thing. Currently, I will show our first steps there. I might not have a lot of time, and we haven't done a lot in that arena yet, but to give you also, just for the picture to be complete, so in that case, bioengineering. So re-engineering the biology we come from via those those inspired or inspirational ideas, re-engineering the biology and make biology do something different. And that would be hybrid life as opposed to life or a life, maybe. Anyway, so, and before that, very brief conceptual introduction. I won't need to dwell on that too much because you are all familiar with complex systems, even though definitions vary a lot. But let me go through these concepts. Of course, I don't need to talk much about this again. This is the touristic brochure about complex systems, the levels of organization of the world, and we all know that what we mean by complex systems, or even though again those words are really ill-defined, those decentralized and emergent and large, what is large, greater than 27, or elementary agents and so on. Okay, fine. So this is what I mean with complex systems, and you kind of already know. It depends on the audience. So I always put this slide first to make clear. And oh, this is a shameless plug about the situation in France. I inserted it for you because you are here a center, and just wanted to show you that in France we also have now these institutes of complex systems that are kind of popping up here and there. The first and the main one is in Paris, but then there is also one in Lyon. And then recently, in the past few years, there have been a few ones also in the province, in various cities in France. And the one I was part of and I directed for 2 years has all these partners. It's a complicated structure in France, but this is not the point of my talk. Just to tell you that we also have Complex System Center, and yet, at the same time, I'm showing you this map, and with a small, let's see, a remark about the fact that these centers generally are more about the science of complex systems, and the social systems, and physics, statistical physics, of this and that, and so on. And there is little in terms of engineering of complex systems, and bioinspired engineering. So this is why I find Southampton here very, very exciting. Anyway, moving on, so continuing with complex systems, every time we have small agents, they get together, molecules form cells, cells form organisms and their brains, organisms form societies, construction or collective motion, and we, specific organisms, this species that we are, with our technology, we form all these incredibly complex societal phenomena that everyone is so interested in trying to understand and bring solutions to, but this won't be our point today. What I am focusing on in this chart, so this is the global, again, brochure, and inside the brochure what I'm looking at, now to the point, is this class of systems that exhibit a strong architecture, a strong morphology, a strong shape, a strong organization. When I say strong, what do I mean? I have not measured anything, but I mean by that, intuitively, I hope you will follow, and we can talk about it. You can, of course, raise your hand and say you don't agree or you don't understand. But it seems that this is the epitome, a multicellular organism like this little crater here is the epitome of an architectured, completely self-organized system, an architecture without an architect, all right? Unlike what a lot of creationists, or all creationists believe, and a lot of Americans, in fact, all still believe. But no, there was no architect, at least not in the sense of putting things together and designing it. And the same goes somewhat at a different scale for example, termite mounds. There is no termite is the architect of the termite mound. Or there is no team of architects, there is no team of construction site managers. Okay, so it's completely decentralized, and no one has the whole blueprint, the whole map of what's going to come up. And yet, it is very sophisticated in its structure, as opposed to a flock. I don't. It was grayed out on purpose to focus on this, but here was an animal flock, a bird flock. A bird flock is random. Okay, so you have worlds and variable densities. There is no architecture in a bird flock. There is no architecture in ant trails. There is no architecture in those famous stripes and spots. In fact, Turing is quoted, or is people think that Turing said, although I never really found the exact quote, but that his famous paper, 1952, on the chemical basis of morphogenesis, but in fact, morphogenesis was a bit of a misnomer, setting aside the fact he was a genius, of course, in many domains, and that was his last work, and he also had a stroke of genius in that case too, naturally, but yet he admitted that he had the stripes, you know, the famous reaction diffusion system, creating spontaneous order out of fluctuations, but that didn't give him the horse around the stripes and the zebra. So I would really separate pattern formation in the statistical sense of repetitive, kind of uniform, even though it's a fascinating phenomenon that we find everywhere, you know, sand dunes and ripples and convection sets and all these things. Beautiful patterns everywhere in nature. Entire books have been published about pattern formation. Sure, fantastic, fascinating, but this is not morphogenetic in the sense of morphogenesis, creating a shape, an architecture. And what do I mean with that? I mean heterogeneity, I mean parts, modules, subparts, appendages, and so on, and most of all, reproducible, programmable somewhat. So if you run again the program, not a very good metaphor, but of a fertilized egg of a zebra, you will get another zebra and the same body plan. Of course, now the details, you know, every single cell that constitutes the zebra is not going to be in the same, they are not going to be the same number and the same place from individual to individual. So sure, you do have an element of randomness at the microscopic level. But at the mesoscopic level and macroscopic level, the organs, the appendages, everything is in place exactly the same from one individual to the next, which you will never get with random striping and random spots. They are never going to be at the same place. All right? So you see what the differences between what I call morphogenetic and non-morphogenetic systems. Anyway, and you know, just to hammer the nail, here is an attempt, also intuitive view of from stochastic pattern formation all the way to very strong morphological systems. And via, for example, here, insects display a bit of all types. You know, again, entrails or a network of entrails are not strongly morphogenetic. However, nests, and especially termite mounds, are strongly morphogenetic. Right? Okay, so this gradient of morphogenesis, very intuitive and waving my hands. And in fact, I take this again, I put it here, already told you the difference between this and that, which I just talked about right now here, excuse me, is that I think the essential difference, and we talked about this at lunch with a few of you, is making the agents stateful. Here, they are stateless, or they don't have enough information to create interesting shapes, morphogenesis, morphologies. And here, if you endow them, if you endow your agents with more information, if you endow, or if they happen to be able to contain more information, then they can create more architectured systems than those random patterns here. All right, so I'm just repeating myself for the umpteenth time, but this time showing what lies underneath is the fact that there is more information. The agents are able to make more sophisticated decisions depending on where they are, their neighborhood, what the interactions among them. But there is another way to look at it, but I won't spend too much time on that. This is the other way where you kind of, instead of making self-organization more informed and more morphogenetic, you do the opposite. You make something that is extremely informed and extremely architecture, but top-down and not at all self-organized. You make it self-organized. Okay, so this is completely the opposite way. But you know, this is another way to look at it. In the middle, anyways, morphogenetic engineering, self-organized architecture or architecture self-organization, it will all end up being the same in the middle, where you have a lot of agents, and agents are sophisticated and they can do various things, and you can get then self-organized systems that are not trivial. That's the whole point. And there is an entire book about it. Second shameless plug, and last one, about the book with 18 chapters. All these people have contributed, just came out a month ago. And you have, of course, a lot of collective robotics in it. People interested in constructing with robots, either the robots themselves cling together, or they bring blocks and build something. You also have simulated robotics here. Hirokisayama's famous swarm chemistry. Yes, this is very typical. So just like wasp mites, you make in that case birds. You give birds different flocking parameters, and you mix different bird species. Okay, it never happens in the sky. You never have different bird species flocking together. In that case, you do here with Hiroki, and you obtain these very surprising morphologies. You know, you have green birds, blue birds, red birds, and yellow birds. Throw them together. They have different speeds, different alignments, angles, and stuff like that, and they form. Interestingly, instead of forming those random kind of masses and clusters, they form some shape. That's very fluid shape, sure, but they form some shape. Things inspired by development. This is my own stuff here. We'll talk about it, and also generative systems, rewriting systems based on grammars. Anyway, all that could be forming what we could call, generally speaking, morphogenetic engineering. All right, and 2 more slides about these concepts, and then show you examples of my own. Let's think a little bit about this, going from the science to the engineering. It has been done before, you could say. It has been done before with neurons, ant colonies giving SEO, genes, of course, evolutionary computation. So this whole bioinspiration doesn't seem to be new. Yet, at the same time, what has happened here is a bit, it's interesting because all these fields here have kind of strayed away from their complex origins. Now, when we also talked about this with Seth over lunch, typically machine learning or, in fact, evolutionary computation have really become fields of meta-heuristics and optimization, and you won't find a lot of, if any, system that is learning or that is being evolved that remain complex in the sense that it is still made of a large number of agents. You won't find a lot of that in the machine learning or the evolutionary computation community. In fact, even ant-colony optimization has become something very much of a meta-heuristics. And where's the origins of the fine-grained multi-agent system in that? And so that's why I think it's important to kind of go back to this. And let's keep that. What I want to do is the same, going from the science to the engineering, the bio-inspired engineering. But this time I take another avenue, and I want to keep its internal complexity, and that avenue is embryogenesis, or you can call it biological development, you can call it multicellular growth, and so on. In fact, there are other communities or families of works that call themselves artificial embryogeny, for example, artificial ontogeny or development, or even evo-devo something. Evo-devo robotics exists now, or artificial evo-devo, and so on. So many different ways of talking about it, but this idea that let's look at the black box of development that has been also, in fact, neglected, interestingly enough, in biology for many decades. Biology is, in the 20th century, the big success was the grand synthesis, the modern synthesis, it is called, between genetics and evolution, between Mendel and Darwin. Okay, let's put them together. We have now, but this modern synthesis has always skipped this step. It was always about correlating the genome with the phenotype, understanding that things evolve because the genome has mutated and so on, but never really looking at this insanely complex, small intermediate stage of it goes through an enormous complex system. So it's not that the genome is coding directly for that foot and that foot. It's not like there is a gene that tells you how long is the limb or the fin or whatever. Of course not. The gene is not saying that. The gene is guiding the small cells in such ways that they are going to form, to divide faster, to adhere in different ways, to migrate differently. And the emergent result is going to be a limb, a fin, and so on, with different structural. But that's an emergent level. It's not directly encoded, you understand that. So this was neglected for a long time in biology. Now it has been reopened, and it's called, of course, biological development, but evo-devo, evolutionary development, is really now looking at it more closely. And if we want to understand evolution, if we want to understand how evolution comes up with all those innovative structures seemingly out of nowhere, we need to understand the building blocks of that. We cannot just continue with a black box evolution. And we need to do the same in bioinspiration, in engineering too. If we want to kind of bring into engineering those ideas coming from biology, we also need to do evo-devo in engineering, I believe. So this is what I'm attempting to do, contributing to, I hope. And I will show you now a few examples. I've been too long on the introduction as expected. In fact, I'm going to go very fast over this one, Julien Delis's work, which is, in that case, trying to be faithful to the biology, trying to be realistic. It's a 3D, it's a fantastic 3D agent simulation of the early stages, not the whole organism, of course, but the early stages of the embryonic development of the zebrafish. Let me get into it right now. And here is in one big picture of his work, simplify the workflow of his work, and then I'm going to fly over the slides because I want to get to the more engineering part and A-Life part. But I still want to show that because it's beautiful. And so, for example, we have raw imaging data, we have microscopy data of a developing zebrafish, and then there is this whole image processing side. Not just Julien, but the whole team that is working on this project. It's called Bioemergence, under the supervision of Nadine Perieras, this developmental biologist I talked about at CNRS. And anyway, so there is this side, which is just about extracting information from the raw images and creating a list of coordinates and velocities and so on. So this is not a model. This is just collecting data, making data structured from unstructured data. But then there is the model part here. How, and in fact, you want to explain this. You want to recalculate this. This is just tracking that, but this is now calculated ex nihilo, right? This is a complete simulation. It's not tracking anything. It's trying to recreate that, even though they don't look the same on this illustration, of course. And the whole point is that you want now to, if your goal is to help the biologist understand, if your goal is biomodeling, then you want to make sure that your simulation is resembles the data you have extracted, and you can superimpose them and measure some kind of discrepancy and validate what you are doing that way. So this is one way of doing it. And this is in very short, big picture, what Julien did. And now I'm going to be very fast. So these are the microscopic images you get from developing embryos. It's you know, you could look at that. You could watch them for hours. It's so fascinating how they they form those structures so fast. Of course, it's accelerated here, but still, within a few hours, you have all these developmental stages. Then this is the tracking side, which is so Julien and myself, we're just part of a big team. It's so much work, so many mathematical algorithms to do image segmentation and so on. Anyway, so this is what you get. And then Julien also did his own, in fact, workflow of image segmentation if it is going to move on. Yes, that's also there are many ways of extracting. Again, in his case, he was interested in vector fields, in displacement vectors, because ultimately he wants to compare his simulated embryo with a real one, and he wants to do this on the basis of vector fields and see if those vector fields are similar. In that case, he will say, my simulated embryo is pretty much like the real one. Okay, that's what he wanted to obtain. So this is this furry ball, this ball of hairs, is in fact a vector field in 4D. In 4D, it's a moving dynamic vector field of the movement that cells create when they start, engulfing the yolk and creating structure. Moving on, so I just breathe. This is impressionistic. Sorry. And then, but I will spend a little more time on this one because it's really the core of not only Julien's work, but also my work with other students on the Bioinspired side. This is the core of how not just me, but basically the way I ended up doing is in my own models of development, how to model development, how to schematize development in a way that can be then programmed more easily, even though of course it is simplified. And for that, I rely on Enrico Cohen's book. Oh, he's not cited here, but Enrico Cohen wrote. He's a plant biologist at where was that? Norwich, in Norwich, yes. And he wrote a popular book about development, and he called it The Art of Genes. And his book is full with artistic metaphors about how an organism grows, an embryo forms itself, and the core aspect is this coupling between sculpting and painting, if you wish. The embryo is painting itself and sculpting itself at the same time. So what does painting itself mean? It refers to the fact that the embryo has a hidden geography. Hidden because when you just look at an embryo under the microscope, you don't see the geography, you don't see the different regions of what? Of differentiated cells, naturally, different cell types. But biologists, in fact, are able to reveal this geography explicitly by inserting markers, chemical markers or radioactive markers, and you can see then, you can trigger those color domains. You can see there is a patch that is blue, another patch that is red. You can mark and dye the embryo in such a way that you can make it reveal those morphogenetic fields, as they are called, meaning that patches of cells have started differentiating and expressing other proteins, right? That's what differentiating means. It means that they have started using other genes and expressing other proteins than the neighbors. So that's why they appear red and the neighbors appear green. So the embryo is painting itself in a covert way, and of course it's sculpting itself. There is the mechanics too. The cells adhere, reshape themselves, migrate, and the tissues that they form fold in very complicated ways. So you have both this painting and sculpting going on, and this artistic metaphor suggested by Enrico Cohen, I think, is brilliant. It really summarizes it all, and you can rely on it then to do your modeling. And this is what I do here on the sculpting side and the painting side. You have cell mechanics and gene regulation, respectively. So here, this is just a little sample of the many ways you can do cell mechanics. So of course, then you have to make choices. You can be more or less detailed in how you model cell mechanics. It can be just, one cell can be a dot, and then you have springs between them. This is very coarse, and yet this is in fact what we ended up doing in most cases. It's already something. You can be a little more refined, and you can say that one cell is in fact a domain of pixels. This is the famous cellular POTS model, if you have heard of it. So a cell is in a domain of pixels or voxels in 3D. You can do tensegrity here. You can do volume, polyhedra, and so on. Many ways of doing cell mechanics. And on the side of gene regulation, there are not many ways there. It's a bit more standardized. In fact, mechanics is probably the biggest mystery. How cells that are neither solid nor liquid, they have a cytoskeleton that they polymerize and then take apart. They have this membrane, which is a fluid mosaic. The whole physics of the mechanics of the cell, their membrane and their skeleton, and the extracellular matrix, the scaffolding between them, is so difficult to understand for physicists and chemists and scientists generally. This is probably now this compared to that, gene regulation, you could say, has almost been understood already. Of course, it's an exaggeration too, but this is more naturally. More people have worked on gene sequencing and gene regulation. The fact that gene produce proteins that will bind to the DNA and in turn regulate other genes. You understand the concept. So that's why you can have networks where you forget the proteins and you have networks of genes regulating other genes. Anyway, so this is the hidden geography here. Okay, those domains of gene colors that reveal proteins and so gene activity. and this is the mechanics, and then so you have this feedback loop, because what is the coupling between both? As cells move, they will change their neighborhood, and so they will change their contacts and the signals they receive, and the signals they receive into this gene regulation network are going to modify the dynamics of it, and are going to paint them differently. Okay, so that's the whole point. And once they are painted differently, once an orange cell becomes dark orange or red, you know, it's starting to sub-differentiate. A precursor nervous cell is becoming more peripheral nervous system as opposed to central and so on. Then they are going to have different mechanical properties. So the color is inducing different mechanical behaviors that in turn is changing the color. This is the whole point. I spent more time on this because it's central to the rest. Now, as Julien is concerned, he continued now his work on modeling. So simple force equations where you have some kind of abstract elastic forces between cells, and you have a dominant neighborhood. Something very coarse, and it's very primitive, and yet it's already very complicated to implement, and it gives very interesting results already. So I'm not going to give you too much detail about this right now. Sorry, I'm going to skip. And this is also a few movies that he obtained. So I've seen this one already. This one is meant to show that he also had to, in his painting, in his case, was ad hoc. It was by hand that he almost literally painted the cells one by one. No, but he predefined the cell types. They did not emerge from the mechanics, but they were scheduled. Okay, you understand? So he took a shortcut there, and he said, here be those, they are called the deep cells here, and then the enveloping layer cells, and then the yolk itself. The yolk is just one big cell, okay? And so on. So he predefined his cells. He had predefined colored cells, okay? But then they had different mechanical behaviors, and that's how he got the whole thing to reproduce somewhat the real embryo. And here is his, this is the methodology again. Last slide about his work. What I showed, you remember this, comparing the real data with the simulated data by just simply overlapping the two vector fields. Those movies don't really show that. Again, it's an illustration, it's a methodological illustration, but this is what he did. And typically he has a simulated embryo there. He can vary a parameter, many parameters of course, but one of them, in that case, the timing of epibole. Epibole is this enveloping like that. And so he gets different simulated embryos as this parameter varies, okay? And then he compares them with the real data. This is the real ball of fur, so the vectors that were extracted from the real microscope images after this very complicated image processing workflow, right? And then he gets, for example, here, there is one parameter that seems to be the most realistic, or let's say the least artificial embryo of all is here. So he gets kind of a peak and says, okay, so in that case, this parameter seems to be optimal and the most realistic one. But then there is the whole question about, okay, what does 15,146 mean for the biologist? Okay, so how do you translate back your simulation parameters into real time and space and other physical units for the biologist to learn something from it? So horrendously complicated, but this was his endeavor. And this was the biomodeling part I wanted to talk about, and now moving on to, from there, moving on to artificial life as such. And in fact, since I have explained already quite a lot about the mechanics and genetic aspect, we find it here again. This was my now old attempt on my own at doing this time artificial development. Okay, so this creature is nothing real, but I wanted to program a proof of concept of what I've been saying since the beginning of the seminar, which is architecture self-organization, which is programmable, reproducible shape that is self-organized. So what is programmed are the agents that self-organize to form this shape, this body plan. And it has, again, it's a bit ad hoc, it's completely empirical, it's numerical. There is not much math behind, if any math at all. I will show you how it works in a few slides, of course. But you know, I think this first model's goal was kind of achieved in the sense that every time I run this is the same movie, of course, in a loop. But if I showed you another movie, you would see the same thing, in fact, emerging. Naturally, again, as I said for the zebra earlier, the exact number of blue cells won't be the same from individual to individual, of course not, and they won't be exactly where they are between them, because there is noise. They kind of jitter around. Yet, what I have hopefully successfully achieved here is that the regions, the mesoscopic regions here that symbolize in an artificial way, abstract way, organs, tissues, and things like that, will be at the same place. And the overall shape will be the same. So that was my goal. I wanted to have a regulated, programmable, and reproducible morphogenesis. So now let me show you what the steps are. And the steps are alternating in a bit, in a contrived way. I'm alternating mechanics and genetics instead of making them play together at the same time. There is a mechanic by division, then the gradient aspect is also important. Let me explain on the next slide instead of here. And then I will show you the latest progress of this. So key to my little model here is a gradient. So you can think of pheromone if the metaphor was insects. In that case, the metaphor is a few scales lower. The metaphor is cells, not insects. But the same idea as pheromone, you have in that case morphogens, or you have molecular signaling, messengers, second messenger signaling, and those things. In any way, it has not been very much clarified in the biology, actually, but there must be some way for the cells to communicate to each other where they are. And this is key in my story here. I need this gradient, you can call it gradient as a shortcut, which is completely decentralized, peer-to-peer propagation of positional information. This is a concept by Louis Wolpert from the late 60s. That, in his case, the French flag, you've heard about the French flag, you know, how cells diffuse some kind of signal, long distance or short distance, and instead of forming stripes as in reaction diffusion with Turing, they in fact explicitly use the information about the level of concentration. So we have this, you know, decreasing curve like this. And if it is between this and that, they will turn blue. If it is between this and that, it will turn white. And otherwise, if they receive very little of it, the trail, they will turn red, you know, the French flag, reading out the position from the morphogen concentration. Very simple idea. Well, same here, except in that case, it's not a real valued decreasing curve, exponential decreasing curve, but it's an integer, more in the computer science way, hopping from agent to agent. So agents are trafficking integers. It's also called gossiping. It's a very robust phenomenon, as you can see. You could already see it many times now. And very simple rules are there. You know, if I'm 4, I want to give 5 to everyone around me. Now, if I am 5 and I receive 4, I will accept it and become 4. But if I'm 5 and I receive 6, I will refuse it because 5 is better than 6. So, you know, you have 2 or 3 simple rules, completely local rules, you know, blindfold rules about trying to send an integer to your neighbors and receiving integers from your neighbors. And that's enough to produce the directed gradients, given, of course, a bit of an asymmetry where you have a source cell, a source of the gradient, which is here in that case, and moves also around. Okay, so not everything is explained. Certainly, you know, again, I made ad hoc choices, but the essential is here. And what do I do with that? Then that's how the painting happens, the self-painting. Why am I green? I am green because I receive 7 from the north source, I receive 3 from the west source, so I have 3, 7, 0, and the rule inside me tells me that if I have those counters, I become green. All right, this is the self-painting. And the self-painting is, of course, the result of, in that case, of a GRN, of a genetic regulatory network. You might not recognize the gene regulation network I showed earlier in the schema of the DNA. because this is, of course, an atrocious simplification of it. But still, there is the idea of a GRN here, of a gene regulation network, where genes are going to trigger other genes and so on, a bit in a feed-forward fashion in that case. But again, it's naturally not the way GRNs really are. But the idea also here, despite all the reservations and the extreme simplification of biology, there is still the idea that there is a GRN that is creating the painting I was talking about earlier, meaning that cells differentiate. They differentiate into red or into blue or into green, depending on where they are. And how do they know where they are? With the gradients they receive, all right? So this picture, this in that case, this kind of checkered board, 3 by 3, is, in fact, you can see that it is scale invariant, it's always there, somewhat, is programmed in the weights of this, by hand in that case, of the weights of this GRN, and self-establishes, okay? So they are self-painting, the swarm is self-painting, but not in a random way, not stripes and spots, in a reaction diffusion way, by reading a program, by executing a program that reads the position, okay? So, and the mechanics, by the way, I didn't even say the mechanics was simple springs, mass spring mechanics. Anyway, so now this is the 3D version, and it's not just for the sheer pleasure of doing 3D, but because we want to do something with those creatures. We just don't want to develop them, but we want to see if we can make them do something in the physics engine that I will show in a few slides. So this was done with Carlos Sanchez in Malaga. Okay, so for now, same thing as in 2D. Same thing as in 2D. So we have a sphere instead of a disk. But the sphere is also segmenting itself into different regions of different colors. Why is it doing that? Because there are 6 sources of gradients. This is just one that is being shown right now, the south one, okay, here. Not diffusing, but propagating those integers that you see here. And this is how cells read their self-established positions and infer their color. Okay, so same idea. You might ask, and you can interrupt me anytime, of course. You might ask, okay, where do the sources come from? Yes, and how are they positioned? Good question. And so, indeed, in the beginning we have them already. We have those 6 sources. So we start with a little ball of 6 cells. We don't go all the way down to 1, but we start with those 6 sources, and then the source positioning is also self-organized. So there are other rules that are specific to being a source, and when you have your flag on, I am a source, then you need to do other things than when you are not a source. And the source kind of go away from each other, so they kind of swim up the gradients, up because integers are growing, the gradients. So south is trying to run away from north, and west is running away from east. Okay, so the sources are also establishing their own spatial positions to kind of span, stretch, unfold like a tent, unfold the coordinate system. All right, so it's completely self-organized. Yes.
Is that kind of cheating the symmetry breaking problem a little bit?
Cheating. Certainly the fact that we have gradients of different colors that are going to be, each cell is going to contain 6 counters is certainly already, by definition, installing asymmetry, because north is going to be somewhere and south somewhere else. Let's see. But I wouldn't call that cheating. It's because precisely the system is not homogeneous. The system, every cell is stateful cells. Stateful cells, they have different information from neighbor to neighbor. They contain those integers that tell them where they are, and so necessarily the whole thing is completely heterogeneous by definition. Okay, anyway, so moving on, then now this is after the body plan, you can grow limbs. This is in fact an image from Enrico Cohen here. So these were only the big areas, the main regions of here be a face, and then you go down into details. This is his way of describing how an embryo actually works is by refinement of details in a multi-scale way. All right. At the same time that the canvas is expanding. So in fact, the scale is always the same. It's the scale of one cell. But since the canvas is expanding, in fact, this canvas is very small. This one is bigger, and that one is even bigger. And so it's completely fractal if you wish, but non-self-similar fractal. Anyway, so I try to do the same here by, now, if you are blue or pink, then you are going to start a new GRN program and start having different mechanical properties if you are blue or green, such that you are going to grow now this kind of plant-like appendage. And so we go away from the blob, from the disk. Because now this is where the painting, the genetics, is influencing the sculpting, the mechanics. The genetics, who you are, where you are, is now feeding back into what you do. Okay? Where you are and who you are is going to affect what you do. And what you do is proliferating faster, adhering differently with other colors, and so on. And that's how you get shape. That's here that you get shape instead of just blobs. Okay? Where you are and what you are will influence what you do. And what you do will take you somewhere, you and your, of course, your offspring, because you also divide. And all that crowd is also going to differentiate into something else, and so on. All right, you see a bit the whole pattern here, and this is the whole genome that is inside every cell. I'm not explaining a lot. Again, there are a lot of details and so on. It's a bit clumsy and complicated, I must admit, but still, and yet at the same time, when I show this to biologists, they of course, they never consider this to be biology. They think this is complete computer scientist craze, insanity, and so on. And when I show this to computer scientists, they say, this is way too biological, way too many details. So I'm always caught in the middle, it's terrible. But for example, here in the genome, those two cells that were kind of pushed hard by, and have themselves divided, of course, so it's a bit artificial to say that this cell did that, but anyway. This is in a real space, they ended up here. This is in genomic space or in execution space of their GRN, their gene regulation network. They have executed different paths of regulation, meaning that this one is a, liver cell, and that one is a brain cell, okay? And they have executed different genes inside the genome. You know that the genome, the big book that every cell is carrying, is in fact used, a cell is using only a tiny bit of it, completely forgetting so many chapters. So cells, as they differentiate, start specializing and looking at just a few pages of that big book and forgetting most of it. Most of it is not used by them, right? So because they say, okay, I'm going to be a nervous cell, so I'm going to focus on chapter 7, and then part A, and then subparagraph B, and so on. I'm going to go in the in that structure of the brain, and so on. So they find their way inside the regulation space of the GRN, and that's how they become different colors and end up in different spaces. So anyway, so this is the 3D version of it. Moving on, and this is the complete movie of a development. And how much time do I have still? Not much, I guess. Started at 4 and it is now 5.
Well, we started at 15:15, so you have 10 minutes?
10 minutes, okay, thank you.
7 minutes?
Yes, I will manage. All right, so this is the 3D. And okay, so why 3D? Now let's move on to something interesting. So in 3D, this is the development in vacuum, or in the egg, or in the womb, or whatever the metaphor is. Sorry, the movie needs to start again. I need to click here. Yeah. So this is the 3D version of what I have explained to you in 2D mostly. Okay, so you got it, there is a body and then there are limbs going out, even though you might not have gotten all the details because I didn't explain all of them, and the idea is that every cell here is carrying the same genome but executing the genome differently inside this set of parameters and regulation depending on where they are, and where they are is the counters they receive from sources. So repeating myself, fine. Now it is hatched, okay, and put in the world. So it's not developing in the world. It develops separately and then it is born and put on the floor. All right. And now we want to test in this physics engine, very simple environment. There is a hardwood floor, there is a gravitational field, and maybe there is an object here, okay, a ball, and that's it. and the rest is the creature itself, which also contains in the 3D case it's not just appendages, but also has a mechanical structure. It has a skeleton and it has muscles. All right. So and let me stop those movie here. Too much animation at the same time. So what we do here, we do two experiments that the PTA would object to probably. Here we dissolve the flesh of the creature, and here we do the opposite. We stripped it of its bones, or in fact maybe we have mutated them in some way. But anyway, this is very easily done. Why is there bone in the first place? Because the gradients say so. There is inside the genomic program, there is also some lines that say, if you are, this is somewhat the central axis of the appendages, not exactly, but imagine this central axis. And the appendages also have their gradients, west and east and north and south and so on. So if you receive about the same counter from west and east, then you must be bone. You must be inside the central axis. Gradients are, you can map everything you want with gradients. So we have decided to differentiate the cells that are somewhat in the central axis, not exactly in that case, into bones, meaning that their edges are no more just springs, but are sticks now. Okay, so we can do that. So the creature is bringing with it those morphological and structural constraints in this world. And the rest, the other edges, and here we really brought ODE to its knees, because we had several hundreds of cells and thousands of edges. It was really panting, and Carlos sent it to a cluster to calculate all that. He was really a wizard in this work. So all these little red edges you see everywhere. Okay, so these are muscle edges. So these are really springs, kind of flexible springs. And these white edges are special edges that differentiated into bone. Okay, fine. So now we put the creature, and here we remove the flesh by just disconnecting all the, saying that all the muscle edges in fact disappear. We can of course do it like this. And here we test actually how the skeleton is resisting gravitation. There is gravitation pulling it down, but it's kind of staying on its own, so it's resisting it, so that's a good sign. And in fact, we have the a contrario proof that the skeleton is also necessary if we remove it, if we do not differentiate the red edges into white. If we keep them all red and all mushy, okay, so it just kind of spreads on the floor. So we do need the skeleton, and that's just to show you very first steps in having fun with those creatures in a virtual physics. Now, how do they move? How do they move here? In that case, we didn't do sophisticated, brain-body co-evolution, or there is not even evolution here. We have designed the genome by hand, by the way. This is a handmade creature. What is handmade is the genome, not the creature, of course. But we have so far designed the genome such that it will have 4 legs and so on. Fine. But it's nothing like brain-body co-development or brain-body co-evolution. The animation part here is completely standard. We just say that the muscle edges that are at the base, the roots of each limb, you can see that each limb is in fact separated into kind of pink and kind of violet here. Also with the gradients, the north-south gradients will tell you if you are pink or violet. Those edges will twitch. Just twitch. Okay, so that's and so it will contract and so make the creature, kind of curve, bend a bit its limbs, and with collision on the hard floor, start moving. This is, naturally happens if you tell all. You put in the genome the fact that if you are a pink cell, then you should also twitch. It's in the genome, so it also brings it with it. So it starts moving and then colliding with this. Fine. So far, so good. And then let me continue. So we have a fitness, a really silly one. Fitness, for example, that's a bad animated GIF, but I will show you something better after that. For example, the creature is fitter if it just manages to move forward and kind of not wind around, but just is able to walk and move forward. So why not? This can be a fitness, but we now run into problems here. I think I put a remark here. Shape reproducibility, which we have achieved, because every time we run this genome, we get the same four-legged creature by development. Doesn't mean, interestingly enough, behavioral reproducibility. So we were surprised that a twin individual from this one, born, simulated in another run, which has the same structure, okay, would walk a little differently and would end up with a different fitness. Because we are venturing now in a very difficult arena, which is the fine-grained complexity. Okay, again, so this individual with a lot of complexity inside it, as I was saying in the beginning. We don't have this a lot in evolutionary works or machinery works. An individual that is internally complex, and so we are fine-grained, and yet at the same time maybe too coarse-grained, because the exact placement of the cells at the tip of the limb might matter in how it kind of tumbles or moves around. So maybe there is a bit of a chaotic effect here. That small variations in the granularity of the tips of the limb might create bigger effects in how it walks. So that's why we took it to the blacksmith and we fitted it horseshoes. And so now we put little horseshoes here to avoid this kind of stochasticity in what exact cell is exactly where and is going to make the creature tumble. So we tried with horseshoes and it was better. So this was without horseshoes. We tried to study also the prints it created on the floor and try to understand what was happening. And then we decided, okay, enough with that, let's put horseshoes. And in fact, also rigidify, it's not just horseshoes, but hooves too. We also added to the genome, hey, purple cells, rigidify, become horn. Okay, become horn. Just don't be muscle anymore. It's too. Anyway, so we tried that. It was better. It was more predictable. It was more that the same species, okay, would walk in the same way. All right. So it was a little better. And so then we were ready to put it in a task that was a little slightly more interesting, less boring than a flat floor. Stairs, for example. And so we asked our creature to, we didn't ask anything, we just started making it animate as it usually does and see if it could climb the stairs. And, so a few movies of that, the horseshoe creature climbing stairs. And these are the first literal steps in exploring, after all this tremendous effort, exploring parameter space. We don't even have evolution yet. We're just very, very beginning of exploring what happens if we vary the limb size. And by the way, varying the limb size only means, in that case, it's developmental, and don't forget, it means stopping the proliferation of cells at a certain point. I didn't even tell you how they stop proliferating. They do that by sending each other a signal and stopping proliferating anyway. So it's all in the developmental dynamics and in the genome that you can regulate limb size, but it's an emergent effect. But anyway, so what if we vary limb size indirectly? We'll get maybe something a little better for limb of size 12, for example. It looks like those creatures are slightly better at climbing stairs. What if we modify body size? So we can have thinner, fatter bodies, longer, shorter legs. You can go from daddy long legs to kind of a big hamburger with a few hot dogs around it. You can. But this is a bit of evolution, but quantitative mutations, quantitative variations around the same body plan. It's always a four-legged thing. We haven't done yet more evolution than that. I have in 2D a few years ago. I skipped that earlier. I have. Oh yeah, in fact, it's here. This is just the 2D experiments that we hope to do in 3D next. We haven't done yet. Where, in that case, mutations are more interesting because they don't just make limbs thicker or thinner, but they add new limbs, or they remove limbs, or they start having different body shapes and body plans. So we can play always with the same idea of a genome inside every element. We can obtain really different architectures. This is handmade examples in 2D. Handmade, the genome is handmade, not the simulation. The genome is handmade. And we hope to do this in 3D soon, and to also make them do interesting stuff and see. But all that is a big endeavor, because this is where I think that this whole exercise, why are we bothering, doing all this? It's huge simulation with a lot of little elements, and for what? For behavior that is not extremely interesting so far in the climbing stairs. But I think that it needs to be investigated further. I really believe that the fine-grained aspect, the internal complexity of the individual is essential if you want to obtain structural innovation. Structural innovation meaning a new limb. In that case, it's it's always kind of limby kind of thing. Okay, so fine. But if you think of real creatures, where did hairs come from? And feathers and scales and a tail and the eye has been rediscovered by evolution many times in different species. Where do all these structures, these organismal structures, come from? Appendages and organs and tissues of all kinds. They come from the smallest building blocks that are the cells. Okay, they are not foreseen or pre-designed by some designer that we are, in the case of artificial devices, and then parameterized. It's not like there may be a hair here, and then it's a matter of optimizing the length. We don't even know that there is going to be a hair here, right? So new features truly emerge from the smaller building blocks. You understand what I mean? The pieces of the puzzle. That's why I think it's extremely important to go through this effort of going fine-grained if we want to break the glass ceiling of evolutionary innovation and let our machines, our designs, find their own substructure themselves that we haven't even thought of. It's also called open-ended evolution. But so you don't have those features that you have thought about in advance. You don't even know, because those features are going to emerge, and I'm repeating myself. Anyway, so this is why I think this effort is still worth trying, and I will stop here. I will not show synthetic biology, sorry, and we have done very little anyway. I just show Susan Stepney's vision of a grown skyscraper. She asked an artist to draw this, and we submitted a project to the European called UComp, Unconventional Computation. We published an abstract about it. Anyway, so this is a bit the same idea of morphogenetic engineering. That is, could bacteria, synthetically modified bacteria, or any other cells, one day could grow this, of course, I mean, a bit maybe like Rachel Armstrong you talked to me about, but this is illustrating the idea that could we steer synthetic biology from just secreting new substance to forming new structures. Okay. Same idea, but instead of simulation, instead of physics engine in your screen, in that case, back to the bioware. Okay, back to the bioware. Reprogramming the real matter. So instead of software, bioware. But same idea, same idea. Anyway, and also wanted to show here this idea about networks, also morphogenesis, but in that case, different space. I'm sorry, I'm rushing on this because it's not, anyway, but instead of 3D, and we're now back to 2D, and we're in fact back to a simpler space, which is cellular automata in 2D. But the same idea that there are gradients being also exchanged between those nodes, those pixels that come one by one, and that is why they are going to aggregate in a specific way. So this is the pure, perfect shape that we want to obtain, and to obtain that, every pixel, they come in one by one, but instead of diffusion limited aggregation, you know about diffusion DLA, right, which is random, and you get those interesting branching patterns. This is program limited aggregation. So not all possible places, not all places are possible. You cannot aggregate anywhere you want. You can aggregate only to where there is an open slot, an open port. And why would one pixel be open to you aggregating to it? Because it has read its gradients and say, okay, I should open my port, and then be ready for someone to come in. Okay? So that's how you get programmable aggregation. Always the idea of programmable self-organization, programmable aggregation here. And then the interesting thing is that we experimented here with David Fouquet about mutating these rules of aggregation and getting, this is the wild type, the perfect shape, and then mutated here randomly. That is, there is true evolution here, true mutation, unlike the other one, there was no true mutation and evolution. randomly mutating the rules that those pixels obey to aggregate to each other, and obtain different species that, in that case, are able. What is the challenge here? The challenge is not to climb stairs. The challenge is to grow around obstacles. This is the challenge here. Grow around obstacles. Those obstacles are little rocks. So the pure one is not able to grow around them because it's completely hard, wired. I mean, it wants to grow there and it cannot go further. Now the mutated ones take the rocks into account and are able to grow around them. And this is another exercise in morphogenetic engineering. In that case, with the added environment constraints that we didn't have very much, in fact not at all, in the growth of the creature. We didn't have any environmental constraints during its growth. We had them later on when it was moving. But here we have environmental constraints while it is growing. And we can do evolution of that too. Okay, so just a brief. And here we go. So thank you to Julien for the biomodeling, to Carlos for the listing, and for Taras who supervised them, and the sequence biology part had very little time. And David and Arsvan were working on this network growth in 2D. Thank you for your patience.